Hello everyone, in this session we will present our systematic literature review paper regarding the effect of image enhancement in CNN-based medical image classification. For the next 10 minutes, we will discuss the effect of image enhancement, starting from the introduction, methods, results, and conclusion. So let's start with the introduction. As we know, through the decades, practitioners and researchers have been developing many techniques regarding image classification. One of them is the convolutional neural network, or CNN, which has been used extensively in different image classification to solve issues in various areas. Despite this, most publications fail to address problems in their dataset quality. One of the problems is the low quality image. As can be seen, low quality images sometimes contain many hidden features that potentially affect the performance of the classification. A solution for that is by performing image enhancement on the images. Some of the methods involved are intensity and contrast manipulation, noise reduction, edge sharpening, and filtering. In image classification, the purpose of image enhancement is to highlight certain image features. Therefore, applying image enhancement during the pre-processing step may affect image classification performance, although the process is frequently used in many proposed image classification techniques. Its effects on classification performance has not been explored in detail. Rahman et al. Applied Grehi and improved the proposed COVID-19 COVID X-ray image classification accuracy by 0.62%. Meanwhile, Freire et al. Also utilized Grehi for the same purpose. However, Grehi does not increase the performance of the proposed model, but rather decreases the precision by 0.1%. Therefore, this paper aims to assess the effectiveness of applying image enhancements in medical image dataset before inserting said dataset into a CNN-based classifier. We hope to clarify how this approach affects classification performance and find the best method for some medical image classification cases.
Next, I will explain the methodology used in this literature review. Next. We aim to understand the image enhancement and classification approaches used in these studies and the effect of those approaches. Therefore, to limit the scope of our research and ease the process of gathering and analyzing information, we formulated 3 research questions. That is, first, what are the problems and related data sets used in medical image classification? Second, what are the methods of image enhancement related to medical image classification? And third, does image enhancement impact positively to the performance of CNN-based deep learning for medical image classification? Next, we searched for publications on 3 internationally accepted databases, IEEE Explore, Scopus, and Science Direct. The search queries consisted of 3 main parts. The first part ensures that image enhancement is used in the publication. The second part aims to find publications that classify medical images. A more general keyword, image classification, was added to gather publications that do not explicitly mention medical as their keywords. The last part ensures that the gathered publications use CNN as the classification approach. Next. After acquiring publications using search queries mentioned in the previous slide, we filter out relevant studies based on the criteria shown in the table. All selected publications must fulfill all selection criteria, but none of the exclusion criteria. Selection criteria are first, internationally recognized academic paper and Scopus index. Second, paper must cover image classification stages. Third, the paper must include image enhancement techniques. And last, the paper must include a comparison between the results of enhanced dataset and the original dataset. Additionally, there are 3 exclusion criteria. That is, first, the papers must be published prior to 2017, especially for the methods. Second one, we do not accept web documents. And last, papers in which the full text is unavailable. Next. So for the selection process, the first selection that is based on abstract and title eliminated 2,237 papers that are not related to medical areas or the classification method is outdated. The remaining 28 papers were analyzed in depth. 12 papers did not fulfill the criteria, while another 7 papers cannot be accessed. The selection produced 9 papers to be reviewed.
After retrieving the papers, we are ready to answer the established research questions. From an overall standpoint, the reviewed papers can be visualized by their publication year. There are 1 paper published in 2018, 3 papers in 2019, 3 papers in 2020, and 2 papers in 2021. Next, problem and the related datasets are discussed in each paper. It is found that there are 6 different problems that were discussed, including breast cancer, diabetic retinopathy, melanoma, tuberculosis, pneumonia, and COVID-19. Furthermore, there are several different datasets used in one problem. Due to this, we would expect that the study can include a wide variety of datasets. Afterwards, image enhancement methods used in each paper are examined. It is found that contrast enhancement algorithms such as CLAHE, histogram equalization, and BCET was used prominently in the papers. Other types of image enhancement such as noise reduction, image sharpening, and color correction and manipulation are covered in at most 3 papers. Furthermore, it is also observed that each algorithm affects image differently. For example, histogram equalization will stretch the dynamic range in the histogram, while Retinex improves the dark region inside an image and equalizes the color tone. Each algorithm has their own use to allow hidden features appear more vividly. Finally, we compared the results when classifying using original images and enhanced images. Overall, we found that most of them improved the performance and succeeded in making hidden features more visible. Some approaches are performed best under certain conditions. Contrast enhancement tends to do well in low contrast images, while color enhancement excels if the task requires color detection. Intensity enhancement is used when features are located in dark regions, and sharpness enhancement is needed when image details are crucial. Despite the successes, some algorithms failed to improve the base performance in some studies. Cherkova and Hardyberg found that automatic enhancement failed due to not contributing positively to color image, while Retinex overexposed the images, causing it to hide more features than revealing them. On the other hand, Munadi used CLHE on COVID-19 X-ray images and found that it creates unnatural enhanced images, worsening the classification performance.
And the next part is the conclusion. This study has reviewed 9 papers to examine the effects of image enhancement methods on the performance of medical classification models. The problems covered in those papers consist of various problems, including but not limited to breast cancer, melanoma, and COVID-19. Furthermore, various image enhancement techniques were used with CLHE and histogram equalization as the most frequently used image enhancement algorithms. In most situations, using image enhancement on the dataset can improve the classification performance of medical images. However, some algorithms fail to perform well than original data on some circumstances. In future works, we would like to investigate whether image enhancement algorithms are needed for a specific medical dataset. In addition, an analysis of which image enhancement method works the best on certain datasets is an interesting topic for future studies. That's all from us. Thank you for your attention.